This method can be used to characterize the physiology of anoxic cells and help answer key questions in host-pathogen interactions, stem cell differentiation, and cancer cell metastasis and therapeutic susceptibility.
This is the first described protocol that enables the long-term anoxic cultivation of mammalian cells.
The implications of this technique extend toward targeted personalized cancer therapy and rejuvenative therapy because due to oxygen toxicity, multiple cell types prefer low oxygen environments for growth.
Though this method can provide insight into cell anaerobic respiration and metabolism, it can also be applied to other systems such as stem cell cultivation.
Generally, individuals new to this method will struggle because extreme care must be exercised to prevent oxygen contamination.
Visual demonstration of this method is critical as the care needed to exclude oxygen contamination is not intuitive.
To begin, add 0.5 milliliters of nitrite stock solution to low glucose DMEM containing L-glutamine sodium pyruvate in fetal bovine serum. Then place 20 milliliters of the medium in a sterile 50 milliliter conical centrifuge tube. Next, degas the medium by placing the tube in a vacuum bell jar at a 45-degree angle. Use a vacuum pump to apply a vacuum at room temperature. Maintain the vacuum for 24 to 48 hours until bubbles are no longer observed. Remove the tube from the bell jar and close the cap immediately to minimize introduction of air to the medium. Then place the tube for 24 hours in a prepared anaerobic chamber and loosen the cap.